Namaskar and welcome to the BSc first year students in this second lecture about microbes viruses. Today we are going to study the general characters of viruses and about the structure of the viruses in detail. Then onwards we will see the recent updates about the viruses. Now the general characters of the viruses. The first and very important character of the virus is viruses are ultra microscopic and submicroscopic nucleoprotein particles found inside as well as outside the cell or the tissue. The second character is they have no cellular organization inside the cell. Hence, they are neither pro or nor eukaryotes. In fact, viruses are intermediate between living and non-living organisms. This character I have already told in the first lecture that the viruses are the intermediate organisms between the living and non-living. The third important character is viruses are exist either intra or extracellular. That means they present in the other organism's body in, that means intra means incomplete structure. They have incomplete structure inside the other organism's body, while whenever they are extracellular they having complete structure. The third important characteristic is fourth important characteristic is when they are found outside the cells they seems to be non-living, crystalline structure like the chemical substance. Whenever they are outside they are having crystal like powder formed structure, or crystal like means diamond like structure of the chemical substance. And then the next characteristic, viruses can be studied in detail with structure by electron microscope. Here we can see the electron microscope, and this electron microscopic structure of coronavirus. Then again we will study with the help of X-ray diffraction method along with biochemical and immunological study, which helps to reveal the antigenic properties of the viruses. Here we can see the X-ray diffraction image of the virus. Then the antigenic for the study of antigenic properties we can get the help of biochemical and immunological study for this study of properties of the viruses. Now we will see the actual size of the virus. In previous lecture we have seen the image in which we have get the idea about the actual size of the virus, which is 100 times smaller than the bacterium. Here is the bacteria and this is the small tiny virus, and which is 100 times smaller than the bacteria and very smaller than the other cell organelles and higher organisms entire cell. Now we will see the examples which are related to the size of the virus. Actually, the size of the virus is ranges between 10 nanometer to the 500 nanometer. But here are some examples which shows the uh, size of the virus. Here, first example is tobacco necrosis virus, which is slight smaller than the ribosome of the higher cell organisms. Ribosome is the cell organelle which take parts in the protein synthesis of the higher organisms cell, and here the tobacco necrosis virus is uh, having size slight smaller than the ribosomes. Then the largest size of the virus is having citrus tristeza virus. Here is the uh, microscopic electron microscopic structure of the citrus tristeza virus, which is having a size of 20,000 nanometer, which is actually the size slight smaller than the bacterium. In previous slide we have seen the shapes of the viruses, either intracellular or extracellular, that they remains whenever they are intracellular they remain incomplete structure, while whenever they are uh, outside the cell of the other organisms, uh, they having a crystal like structure or complete structure. Now here are some the pictures of the viruses from which you can get the idea of about the shape of the viruses. Some viruses having rod shape, some having spherical shape, some are having brick shape, then some are having bullet shape, then some are pleomorphic, some having a tadpole like structure. Here we can see the image of bacteriophage, which is having a tadpole or spider like structure. uh, there are many other examples of the viruses. Here the common example or very important example of HIV virus, which is having a spherical shape, hepatitis B, Ebola virus, then adenovirus, influenza virus, rabies virus, herpes, rota, papilloma. There are many uh, viruses which are having different shapes. Now regarding the structure of the virus, how the structures or how the viruses are look like, we will see in this slide. First of all, it is mainly consist of central core nucleic acid. Here in this three-dimensional image of the virus, in which uh, it shows crystalline structure of the virus, which shows the core part of the or central part of the virus, which is contains the nucleic acids surrounded by protein coat. Here is the protein coat around the nucleic acids of the viruses. In some cases, the viruses having envelope around this protein coat or surrounding the entire body of the virus, which is called envelope around the body of the virus. Then they contain genomic nucleic acid molecule, which may be monopartite or monopartite, bipartite, tripartite or multipartite. Monopartites means it contains only a single molecule of genetic material, nucleic acids. Then capsids protect genetic material from nucleases and other enzymes. Further, it helps in the process of transfer of viral nucleic acids between the host cells. So, the capsid, which is surrounded to the nucleic acids of the viruses, they protects the virus from the nucleases and some other enzymes present in the higher organisms body or higher organisms cells. uh, Similar, again they help the envelope or the capsid, sorry, the capsid helps to transfer the genetic material, the nucleic acids from the virus to the higher organisms cell. Then each capsid is made up of several capsomers. Here we can see on this entire body of the virus, the capsid is made up of several capsomers. Here is the capsomer, and capsomer is made up of subunits called the protomers. Here are the small spherical structures. Each single structure is called protomer, and the group of 5 to 6 protomers combine to form a capsomer. Then the few genes codes for the proteins that assemble to form a capsid. In the genetic material having genes which codes for the assembling of the proteins to form a capsid, the capsomers symmetrically arranged around the nucleic acid core, which is in different forms. On the basis of symmetrical arrangement of the capsomers on the body of the virus, we will see different types of the symmetry of the capsomers on the entire body of the virus in next lecture. Due to the pandemic situation by the coronavirus, we started to know the ABCD of the virus, and we get idea that not only the coronavirus, other than coronavirus, there are many viruses which are harmful to us and the plants also. So the recent update regarding the virology or virus world is the Nobel Prize is given to the physiologist or scientist in medicine in this year 2020 to the Harvey Alter, Michael Houghton, and Charles Rice for the discovery of hepatitis C virus. As they discovered hepatitis C virus, we must know the entire idea about hepatitis A virus, hepatitis B virus, and the recently discovered hepatitis C virus. In next two slides or two videos, we can get the entire idea about hepatitis virus. So please watch the next two videos very carefully and get the recent updates regarding the hepatitis virus and Nobel Prize of the regarding the hepatitis virus.
If you have hepatitis A or B, your liver is inflamed because you have been infected with either the hepatitis A virus or the hepatitis B virus. Your liver is a soft, flexible organ that performs many important functions. The functional parts of your liver are called hepatic lobules. Your hepatic lobules filter all of the blood in your body. As the blood passes through, your hepatic lobules break down harmful substances. Remove bacteria and worn-out blood cells, and forms clotting factors that control bleeding. After a meal, your liver stores nutrients to provide your body with energy when needed. Your liver also makes a substance called bile. Your gallbladder stores bile and releases it into your small intestine to help digest fats in the food you eat. If you have hepatitis A, the virus entered your body when you were exposed to fecal matter from a person infected with the virus. You may have been exposed to the virus by eating contaminated food or water, contact with infected feces, such as during a diaper change, or having unprotected sex with a person infected with the virus. If you have hepatitis B, the virus entered your body when you were exposed to the blood or other body fluids from a person infected with the virus. This could have happened from sharing a drug syringe with an infected person. Other ways you may have been exposed to the virus include having sex with someone infected by the virus, sharing personal hygiene items such as razors or toothbrushes used by an infected person, direct contact with the blood or body fluids of an infected person, or when a mother passes it to her baby during birth. When either the hepatitis A or B virus enters your liver, it invades your liver cells and makes copies of itself. In response, your body sends immune cells to attack both the virus and liver cells infected with the virus. As a result, these liver cells become inflamed and then die. Over time, scar tissue forms around dead and infected liver cells, which prevents your liver from working properly. If you have a chronic hepatitis B infection, your liver contains a large amount of scar tissue called cirrhosis, which limits blood flow and results in permanent shrinking and hardening of your liver. If you have hepatitis A, your doctor won't prescribe any medical treatment because your immune cells will eventually find and destroy all of the hepatitis A viruses in your body. If you have hepatitis B, your immune system will usually remove all the hepatitis B viruses from your body. In some people with chronic hepatitis B, particularly children, their immune cells are unable to remove all of the hepatitis B viruses. If you have chronic hepatitis B and your immune system can't get rid of the virus completely, your doctor may prescribe antiviral medication. If you have a severe case of chronic hepatitis B, your doctor may recommend a liver transplant operation. If you have hepatitis C, your liver is inflamed because you have been infected with the hepatitis C virus. Your liver is the largest organ inside your body and performs many important functions. The functional parts of your liver are called hepatic lobules. Your hepatic lobules filter all of the blood in your body. As your blood passes through them, your hepatic lobules break down harmful substances, remove bacteria and worn-out blood cells, and form clotting factors that control bleeding. After a meal, your liver makes and stores nutrients to provide your body with energy when needed. Your liver also makes a substance called bile. Your gallbladder stores the bile and releases it into your small intestine to help digest fats in the food you eat. If you have hepatitis C, the virus entered your body when you were exposed to the blood or other body fluids from a person infected with the virus. This could have happened from sharing a drug syringe with an infected person. Other ways you may have been exposed to the virus include having sex with someone infected by the virus, sharing personal hygiene items such as razors or toothbrushes used by an infected person, direct contact with the blood or body fluids of an infected person, or when a mother passes it to her baby during birth. During the early or acute phase of a hepatitis C infection, the hepatitis C virus enters your liver and invades your liver cells. Once inside your liver cells, the virus begins to make copies of itself. During the copying process, changes called mutations occur frequently in the virus's genetic material, leading to new strains of the virus. In response to the viral infection, your body sends immune cells to attack both the virus and the liver cells infected with the virus. As a result, these liver cells become inflamed and then die. Over time, scar tissue forms around dead and infected liver cells. The scar tissue prevents your liver from working properly. If you have a chronic hepatitis C infection, your liver contains a large amount of scar tissue called cirrhosis, which limits blood flow and results in permanent shrinking and hardening of your liver. A vaccine contains weakened or inactive viruses that train your immune system to recognize and attack certain viruses. However, frequent mutations in the hepatitis C virus make it likely that you will catch a version of the virus not contained in a vaccine. As a result, there is no effective vaccine for the hepatitis C virus. If you have hepatitis C, your immune system alone may destroy all of the hepatitis C viruses in your body. However, most people infected with the virus develop chronic hepatitis C, a condition in which your immune system is not able to destroy all of the viruses in your body after 6 months. If you have chronic hepatitis C and your immune system can't get rid of the virus completely, your doctor may prescribe a combination therapy consisting of an antiviral medication called ribavirin and injections of interferon, a medication that strengthens your immune system. These drugs are most effective against hepatitis C when taken together. If you have a severe case of chronic hepatitis C, your doctor may recommend a liver transplant operation.